Good morning, dear colleagues and friends. So let me introduce my projects today. My name is Gantsetseg Garmaa, as you may know. My thesis title is The Role of miRNAs in Renal Fibrosis and Their Molecular Pathways. You can see our team here, my supervisor, an SMS, and biostatistician. And in brief, my vision is to contribute to build the healthy communities which may be supported by evidence-based and also translation medicine. And during my PhD study, my vision is to investigate the novel microRNAs that may play an important role in kidney disease. And regarding my vision in PhD, I set two specific goals, which means like two projects. And the first one is investigate the most dysregulated microRNA signatures in mammalian chronic kidney disease from the profiling studies, which means we're going to deal with like thousands of microRNA lists in the first project. Then second project is I was interested in investigating the diagnostic accuracy of already published microRNAs in kidney disease. And let me introduce the first project in detail. The title is microRNA biomarkers in early and late stage of chronic kidney disease, meta-analysis of human and marine profiling studies. You can also see our team here. And it's the second submission. We were submitted to Journal of Translational Medicine and waiting for the response now. And the first project, before I introduce the results, I would like to a little bit introduce about the background and why I decided to choose microRNA in kidney disease. As we all may know, the chronic kidney disease is the silent killer, I would say. And the 10% of the world population is affected by kidney damage. And those people can lose up to 90% of kidney function without experiencing any severe symptoms. And then this is really awareness, that's really increasing the awareness of this kidney disease management. And I think like maybe microRNA might be the potential diagnostic microRNA to investigate or find out this kidney damage. And these non-coding small RNAs have a potential to target the gene, also affect the protein expression of any kind of disease. And then if we look at the individual microRNA function, it has a lot of interaction with the genes and also proteins. It can activate the genes, also repress the genes. Also, it can interact with the chemical drugs and substances. And also, it's also play an important role in pathways in any disease. And then, before we start this project, just listing the name of the microRNA is not enough. And then we also have to investigate the target genes, also molecular pathways in kidney disease. That was the initial set point here. And then we asked the question, which are the most dysregulated microRNAs in human and animal security? We also choose the animals because thousands of scientists are working in animal, and then we wanted to see the difference, what's happening in human and animal. Then we set the PICO framework. The P was humans and animals tested for microRNA profiling studies, which means these studies has to be tested thousands of microRNAs, and then exposure of persons of security. And the comparator was mostly healthy individuals and animal model, and the outcome was most dysregulated microRNAs by ranking order. Then we hepatized the CKD, on the other hand, kidney disease, characterized by dysregulated microRNAs. And then maybe introducing the meta section might be also interesting for you, and also give you the hint about understanding my results. And the first, let's say now we have like over 2,000 microRNAs registered, and there were thousands of articles, of course, in PubMed and any other source. And then we were only interested in microarray or gene sequencing and other multiple well-played PCR techniques. And then we collected data around 17,000 articles, ended up human 32 and animal 13 study. And after we collect the articles, we put the all microRNA list using robust string aggregation and like this. And then after that, we ranked them in Excel. Then we performed the robust string aggregation itself in our program. Then after that, as I told you, we determined the target genes, also molecular enrichment analyzes using bioinformatics tools. And now it's time to introduce the results. The results mainly came from the robust string aggregation analysis, and most important results will be up and or down regular microRNAs. There you can also see subgroups by human or animal or different kidney disease, and also sample types, urine or blood or kidney tissue. And in summary, the subgroups will look like this. For humans, 3 main, and animals, 2 subgroups. And this is the also example of our results. This heat map will show you in the x-axis, these are the individual studies, and the y-axis you can see whether this individual microRNA is significant or not. If it's like looks like more reddish, it says significant results. And so in mice kidney tissue, there were 6 microRNAs were significant dysregulated in kidney models, disease model. And in the summary results, actually we got similar results for each full and subgroups, and this is the summary of my results. In human CKD, in a urine sample, there were 4 microRNA were down, 1 is up. In the blood, 2 is down, 1 is up. In the kidney tissue, only 3 microRNA were down regulated. And mice diabetic kidney model, 2 is down, and a UUR model is the renal fibrosis model. It's like 5 microRNA up, 1 microRNA were down regulated. And then after we got these results, we also checked all individual microRNAs, whether it's validated by PCR results like COVID taste, and then 2 microRNA were not validated. It was not also published. And then now we are doing a PCR and also working with the human samples to investigate these microRNAs and also validate these microRNAs. And also, we were interested, okay, there's like several microRNAs up and down. What's going to happen in early stage of kidney damage or also in the late stage? And there, in early stage, interestingly, there were only 2 microRNA was up, and also in the late stage, most of the microRNA were down regulated. That was also quite interesting for us. And then as I also introduced, we checked the target genes, and then I just tried to put the sample types here in the urine figures, which means these microRNAs came from urine samples, and the numbers are number of target genes. And there is thousands of target genes, and we cannot really say like this microRNA is targeting specific genes, and after that, we tend to perform the molecular pathway analysis to understand what's really happening behind this microRNA. And then this is the example of molecular pathways. It will call like enrichment analysis from DynaTools. And this is also from animal kidney tissue. And most dysregulated molecular pathways were fatty acid and prine, ECM receptor interaction, and steroid biosynthesis pathways were most dysregulated in kidney tissue. And in conclusion, we were investigating sample-specific, for example, urine, blood, and kidney tissue microRNA signatures, and chronic kidney disease, and also stage-specific, like early and late. And also, we were trying to just investigate what is happening in translation of animal model to human side, and then investigated the molecular pathways of these microRNA signatures. And we can also call a simple study implication here. In a clinical side, we were bringing up the candidate microRNAs in a clinical investigation further on. Maybe we can check this microRNA list in a clinical setting like we are doing now. And also, the sample-specific microRNA is quite important in there. And maybe we can improve the diagnostic approach further on. And also, strand and limit, while we are working with the data, there is lack of raw data was common, and then it was quite complicated to perform an analysis, and that's why we decided to have robust string aggregation. And for animal models, we only included the mouse data because of the availability of the data. And my second project is the diagnostic accuracy of microRNAs in chronic kidney disease. In this project, we are only focused on human chronic kidney disease. You can also see our team here. And for this project, the background will be pretty much the same. We just wanted to see what's the diagnostic accuracy of these published microRNAs. And the question was, what is the diagnostic accuracy of microRNAs in humans diagnosed with CKD compared with the healthy individuals? And then we set the peer framework. The P was healthy, and then CKD humans. E index test was microRNA detection by only PCR, which is like more validated tests. And reference test was biopsy or clinical parameters, which means the patient has to be only diagnosed by kidney disease. It has to be proven, and the diagnosis was CKD. And there is a clinical implication. So let's say there's thousands of articles, but we're hoping that this is the second meta-analysis in the science, and then maybe we can just see the diagnostic accuracy of microRNAs in individual kidney disease. For example, the previous meta-analysis was only combined all the CKD, but in here we were trying to separate all kidney disease. And you can see the systematic search and numbers and search key here. And this is the progress of the second project. We finished the data extraction and we have 100 articles, and then we finished risk of biases and then waiting for the results now. And this is the summary and a planned date for submission. And I will end up my talk like saying Mongolian proverb. It means which means where there's a will, there's always a way. And thank you for your attention.
Thank you. It was a really nice presentation. I just wanted to ask where would you put this microRNA methods? To rather a screening method or a direct diagnostic method?
I would say it would be more individual diagnostic, because microRNA test is quite expensive, and just screening for the whole population is not that. Wouldn't that make sense? Also, I think it could be accurate enough to diagnose, perform the test in individual patients.
Thank you so much.
My question is that now you are, quite experienced on this, that when you compare them, although the human data are not quite ready, are the same microRNAs changed in humans and in mouse, or these are different? This could be species difference, and also this could be due to the different, animal models always have some kind of shortages compared to the human disease. So do you know this already?
Yeah, for animal model, they were only tested microRNAs in kidney tissue, and then we found several similar microRNAs that present in mice and also human, but it was not 100%. Also, considering the animal model of the kidney disease, it's quite different than human original disease. That might be the reason too.
The last one, which is related to Gabor's question. What do you think, which animal model would be more suitable than the mice model? Because you mentioned that only mice model was considered. Do we have any knowledge that in terms of microRNA assessment, which animal model would be better than the mice?
I think the UUO model, it's a chronic kidney disease model. I think that would be the better one, because most of the results were matched with human results. It's a kind of surgical method. They were just kind of cut the urethra, and they will just damage the kidney on purpose. And so they will wait for 2 weeks and figure out what kind of renal fibrosis is present in the kidney tissue. And I think that model might make sense.